And let us see now an animated video.
The complement system. The complement system, or just complement, it is a set or group of plasma proteins, and these proteins are made constitutively by liver. These proteins are present in blood, in lymph, and in extracellular fluids. The complement system is so named because these proteins assist or complements the immune cells in destroying pathogens. Most of these proteins are proteolytic enzymes. In the absence of infection, they circulate in inactive forms. When there is an infection, these proteins become active, and this is known as complement activation. That is, when inactive proteins become active, it leads to complement activation. One important property of complement protein is that they act in a cascade, which means one reaction stimulates another reaction, which further stimulates another, and so on. The complement activation leads to three major consequences which help in eliminating the pathogen. These consequences are enhanced phagocytosis, inflammation, and cytolysis. We will study each of these consequences in detail later. There are three complement activation pathways, and these pathways differ in how these pathways are initiated and which proteins are involved. So these pathways are the alternative pathway, the lectin pathway, and the classical pathway. Before going into details of these pathways, it is important here to first understand the nomenclature of the complement system. So let us first study nomenclature of the complement system. The enzymes of the classical pathways were identified first. They are designated by capital letter C followed by a number. These numbers indicate their order of discovery. There are 9 such proteins, C1 to C9. In complement system, when a protein is cleaved or broken down into two, the resulting peptide fragments are designated by adding a lowercase suffix, that is either A or B. Suffix A represents that this fragment is the smaller one, and suffix B represents that this fragment is the larger one. For example, complement protein C3 is cleaved and these two fragments are produced. The large one is represented by C3b and the smaller fragment is represented by C3a. Now, it is generally the larger fragment, that is, the fragment with the suffix b, which contributes to next enzymatic activity in the cascade. Thus, in this case, C3b will contribute to next enzymatic activity. Complement protein C2 is an exception. How? Because in this case, the larger fragment is C2a, not C2b. So when complement protein C2 is cleaved, the larger fragment is C2a and the smaller fragment is C2b. We said that in the classical pathway there are 9 proteins, C1 to C9. Now here again there is one exception, that is C1. Because C1 is not a single protein, it is a complex. It is a complex of 3 proteins, namely C1q, C1r and C1s. Proteins of the alternative pathway are termed as factors, and they are designated by capital letters, for example, factor B and factor D.
Now seen a wonderful this animated video wherein the details of classical pathway, alternative pathway and lectin pathway are shown. Some of the what is it called complement proteins are shown. A few of the complementary proteins are getting cleaved. As a result, C3b is a big component, C3a is a small component was shown. So there are several aspects that are being involved. Let's go and see the details now. So the objectives of this program, this particular lecture topic is to understand the serum dormant proteins involved in innate immunity. Serum dormant proteins involved in innate immunity. The second aspect is to appreciate the role of complement proteins in lysing bacterial cell. Ultimately, the function of the complement protein is to lyse the bacterial cell. How does it lyse? That's what we have to understand now. Now, the expected outcome of this particular topic called the complement system is as follows. The complexity of the serum dormant proteins involved in lysing bacteria will be known. It is not simply one complement protein comes and makes the bacteria to get lysed. There is a series of cascade of events. That cascade of events we have to understand. Why to understand is if there is any deficiency in this cascade of event, a person, a host system is becoming ill. Okay. Now, the deficiency of complement proteins that cause diseases will be known. So, these are the two expected outcomes that we anticipate from the learners after the completion of this topic, they will get familiarized. Now, let us see. As I was mentioning to you, this complement topic is a Nobel laureate topic. He is Jules Bordet, and he secured the Nobel laureate. He was awarded with the Nobel laureate this in the year 1919. And he proposed this. These are the complement proteins. How did he say these are the proteins? They are all getting, the proteins get inactivated. The proteins get inactivated. The proteins get inactivated. Oh, rather. The proteins get inactivated at 56 degrees centigrade. Starting from 45 onwards, they get inactivated. The complement proteins are inactivated at 56 degrees centigrade for 30 minutes if it is heated. That's what he has come to the conclusion. They are the complement serum proteins. So let us see by having this complement system, what is the advantage for the host? Opsonization to enhance phagocytosis. So we have to understand the opsonization. I'll give you a simple example. A few biscuits small children do not eat. Along with the biscuits, if there is a cream that is put on the biscuit, for the purpose of this cream, the whole biscuit is being eaten by the small kid. So that is what here called opsonization. So I will draw a small diagram. Let us see here, there is a bigger parasite. So if it is a bacteria, if it is a virus or macrophage engulf. If it is a bigger parasite like trypanosoma, if still there are some more bigger parasites, our macrophages will not engulf. So this any organism, they are having on the surface proteins. These proteins are called antigens. So any parasite, for that example, if you take up, they contain the proteins on the top, they are said to be the antigens. Okay. Our system develops immunoglobulins against antigens. These are the Y-shaped structures called immunoglobulins. That was the first chapter that we have seen in this unit 2. So these immunoglobulins bound to the specific to the antigen. Okay. Once these immunoglobulins go and occupy the respective position Fab, that is fragment antigen binding, then this immunoglobulin, this portion of the immunoglobulin is called CH3, that is constant heavy region 3. It is not the chemical equation, it is constant heavy region 3. So all of them are having constant heavy region 3. Okay. So the constant heavy region 3 is there is a receptor in most of the cells for constant heavy region 3. So, the cells are getting accumulated, the cells are being attracted towards the back. This is what is called bacteria. Okay. The cells are attracted not because of the bacteria, they are attracted because of the immunoglobulins and CH3 receptors. So, they release the granules and see that the bacteria dies. Here this portion is called opsonization. So, that is what opsonization enhances the phagocytosis. Phagocytes attracts and activates, and lysis of bacteria and infected cells keep going on here. And then regulation of antibody response takes place. And then clearance of immune complexes. That's what whenever a person is having regular infection, he is having all over the body immune complexes. What is immune complex? Let us see now. So, let us say this is an antibody. This is an antibody. Why I am showing antibody as a star? Because antibodies are all folded, proteins are all folded, antigens are all folded. This is an antigen. Antigen are all folded. Okay. So, once there is an antigen, for this antigen there is an immunoglobulin developed. So, okay. Now, in the center there is an antigen which is shown as star shape. This antigen is having several epitopes, number 1 epitope, number 2 epitope, number 3 epitope, number 4 epitope, number 5 epitopes are there in this antigen. For each of the epitope, there is one, this is what antibody made against epitope 1, antibody made against epitope 2, against epitope 3, against epitope 4, against epitope 5. So, this is now called the immune complex. Okay. So, such type of the immune complexes get cleared by this type of the complement system. So, clearance of apoptotic cells. Okay. So, what is the detriment to the host is it causes inflammation, it causes anaphylaxis. Anaphylaxis is instant allergy reactions. Okay. Now, let us see what are all the proteins present in this. So, if you take up earlier I told you more than 20 types of serum dormant proteins are there. They are all named as C1QRS. The QRS also we have seen in the video. C1QRS, C2, C3 to C9. They are all named as like this nomenclature. Apart from that, factor B, factor D, factor H and properdine. Properdine pathway is most important. And mannose binding proteins. So, several proteins are there. They go beyond 20, just beyond 20. Okay. Now, as we have seen that these proteins are all dormant, they get activated. Once they get activated, they are given a overline. They are given over, this is the overline is given on that. Okay. So, that is what happening. This just like that. They are given overline. Just that is for our sake of understanding. In the regular system, there is no overline, there is no such type of these things for us to understand. Okay. Now, let us see. So, now when it is getting cleaved, when these components C1 to C9 getting cleaved, the larger component there is one, there is smaller component another. So, the larger component is being referred by the larger component is being referred by is being designated as B. Smaller component is being designated as A. Only an exception is there with regard to the C2. Okay. In general, the larger component is B, smaller component is A. So, the name is A given in such a way that the smaller component is going to become a part of the anaphylactic reactions. Okay, that is what. So, there are in the video also it is shown there are 3 pathways. One is the classical pathway, the next one is lectin mediated pathway, the other one is alternative pathway. The 3 pathways. Okay. So, the 3 pathways are not totally independent, they are converging in between. Let us see. So, it is antibody dependent. The first one, classical pathway, is antibody dependent. In the previous slide, I have shown you the immune complex. The immune complex is nothing but antibody binding to the antigen. That is what antibody, that is, it depends on that. Next one is these 2 are antibody independent. Okay. So, this is what the stage where all the 3 get converged, activation of C3. So, once this is activated, C3 is activated, there is the generation of C5 convertase. That's what I've been telling to you. There is C1, C2, C3, C4, C5. Okay, so C5 is now getting converted. So how does it get converted? It's going to become C5b and C5a. C5b and C5a. Okay, so that conversion takes place. As a result, hereafter, C5b comes into the scene. So now activation of C5a, lytic activity pathway starts. Lytic activity pathway starts. Okay. Now let us see. So these are all the various components of the classical pathway. As we have seen earlier, classical pathway, alternative pathway, lectin pathway. So these are all the various components. This also we have seen in the video, C1QRS complex. Okay. Then C2, C3, C. For the sake of us to understand, they're all given in different shapes. Now let us see. So this is an important aspect we have to understand. Okay. So the line that is given here is not just like that. It is drawn. It is nothing but bacterial cell membrane. Bacterial cell. This is a bacterial cell membrane. Okay. So every organism on the top they are having antigens. This is an antigen. This is an antigen. So bacterial cell membrane is shown, antigen is shown. Now let us see what is happening. Afterwards, immunoglobulins. Wherever there is an antigen, immunoglobulins come. So this is called an immune antigen, antibody, and immune complex. Okay. Once the immune complex is formed, C1QRS complex is getting attracted. C1QRS complex is getting attracted. Once this is attracted, then C1QR is getting activated. What is the activation here? It is going to become enzymatic. What is the enzymatic? It is going to become C4 convertase. Let us see. Now C4 comes. C4 is getting converted as C4b and C4a. So the C4b rests on the bacterial membrane. So let us C2 is again, it is a converter of the C2, C2a and C2b. C2a comes here. That is an exception C2. That's what shown in the previous slide. So now this C4b, C2a is C3 convertase. That is, as we have seen previously, cascade of events taking place one after the other. Okay, so C4b, C2a are C3 convertase. That is, there is one that is coming to the scene now. That is called the C3. That C3 need to be converted as C3b. Let us see now. C3 comes. C3 has become B now. Okay, so now C4a, C2, C4b, C2a, C3b. This is now called C5 convertase. Okay, with this the classical pathway ends. Okay, now it converts C5 into C5 A and B, C5b rests later. Let us see hereafter. So these are all the various biological activities of C3A. So C3A, let us see, it is anaphylatoxin. Okay, so it causes, it influences the basophils and mast cells to produce histamine. So C3b does opsonins. Opsonin promotes the phagocytosis, and C4a is again anaphylatoxin. Anaphylatoxin means it influences the basophils, mast cells to produce the histamine, a sort of allergy reactions. C4b is again opsonin. So now let us see the components involved in alternative pathway. Hitherto we have seen the components involved in the classical pathway. That is what C1QRS, C2, C4, C3 we have seen. Okay, ultimately we ended up there with C5 convertase. Here also we will end up with C5 convertase. So in alternative pathway there is C3 is again comes here, B factor comes here, properdine factor. All of you remember this is the properdine factor, that is most important aspect in the complement system. The deficiency leads to some illness. Okay, now these are the various comparison between the classical pathway and alternate pathway. So this is C4b, C2a, C3b, that is one combination to convert C5. Here in alternative pathway C3b, BB, we have seen B factor, H factor, properdine factor. There again it becomes BB and C3b. This is again the C5 convertase. So both the things terminate at C5 convertase. Now let us see the lytic pathway. It is most interesting, where we are going to see the formation of the perforin molecules. So in the lytic pathway starts from C5 convertase. C5 is the starting point that is going to get seated on the bacterial cell membrane. So now these are all the components involved in the lytic pathway: C5, C6, C7, C8, C9. Okay, let us see now. So now, as I have shown you earlier also, this is a bacterial cell membrane. So bacterial cell membrane, now our interest is to perforate, to make holes on the bacterial cell membrane. Why to make holes on the bacterial cell membrane? This is one type of the defense. This is one type of the defense mechanism that the host system is having. If you make the perforations, bacteria oozes out, so then the bacteria no longer survives. Okay. So now let us see C5. C5 is going to become so in the previous case, this we have seen C4b, C2a, C3b is C5 convertase. Okay. So once now the C5 comes, it is going to become C5b. C5b sits. C5b attracts C6, it attracts C7, it rests on the bacterial membrane. Now C7 attracts C8, C8 attracts C9. Okay, C9 means it is a complement protein 9. Okay, now this is the bacteria on this. So this is what the perforation that is made here. Okay. So this is what the perforin molecule. So with this, the three types of what is it called, these pathways, as we have seen, the classical pathway, lectin mediated pathway and alternative pathway, terminates at the formation of the perforin molecule. So let us see some of the biological properties of C activation products. So C3b does opsonization, C4b also does opsonization to promote the phagocytosis. Now there are some of we have listed more than 20 molecules here. Let us see the deficiency. See, all the molecules are not present in everyone. A few of us are ill. A few of us are getting periodically bacterial infection. Why it is? Because there are complement proteins are deficient. Let us see a few now. So in this case, there is C1 inhibitor, that is complement 1 protein inhibitor. If such type of the people will be having, if there is deficiency with C1 inhibitor, he will be having hereditary angioedema. Hereditary angioedema means the blood, hereditary angioedema means here the blood vessels, all those things are got, what is this called? That is angioedema. That means more number of blood vessels are formed, so that region is become bulged. That is, bulged means a sort of expanded. So that's called edema. The term edema being used. So there is a SLE, predisposition for SLE. Here SLE means there is a situation called, this is a butterfly type of arrangement here that comes. So that's what because of this deficiency C1, C2, C4, complement 1, complement 2, complement 4. If they are deficient, there is a situation called systemic lupus erythematosus. Systemic lupus erythematosus. This is an autoimmune disorder. Okay, this autoimmune disorder leads to such a type of disease that just now I have shown you, butterfly type of the shape of the inflammation on the face. Okay. So that's what. So now still let us see the deficiencies. This is mannose binding lectins. If it is deficient, we are going to have bacterial infections, and factor B and D, if they are deficient, in all the cases pyogenic bacterial infection, we are susceptible to the bacterial infections. That means whomsoever is having more of bacterial infections means they have got complement proteins at a deficient stage. So here there is another important, beginning I have been telling to you, properdine. Okay, if the properdine is X-linked. X-linked means it is more being expressed in males. Females are having the 2 X chromosomes, male is having 1 X chromosome. Unfortunately, if it gets inherited, this properdine deficiency system gene, then it is being expressed in the males. Okay, once if it is being expressed, it causes it causes a susceptibility to meningitis, meningococcal meningitis. Meningitis means for the brain, there are membranes in the brain, those membranes are getting infected with Neisseria meningitidis. These are all the membranes that are present in the brain, they are getting infected with Neisseria meningitidis. Okay. Now let us see. So now let us see the summary. Complement serum proteins are dormant. That's what we have been talking since the beginning. They are involved in innate and humoral immunity and humoral immunity, humoral innate immunity. So with this summary and the details that you have seen in the cytokine chapter topic, let us go through a few of the assignments or study questions. Now the first one is define complement system and enlist the components of the complement system. The third, in brief, explain the classical, lectin and alternative pathways, and explain the complement system up to the generation of C5 convertase. Okay. The next, how does the generation of C5 convertase leads to activation of the lytic pathway. Okay. The last one is enlist at least 5 complement deficiency diseases. Okay. Please come prepare with this. With this, I thank you all for your patient listening.